Lucy is a lecturer at the Institute of Psychiatry, Psychology and Neuroscience, having done her PhD there in anti-psychotic response, and went to UCL, where she worked on several postdoc positions, but is currently doing work on how brain development relates to mental health. And so she's the perfect person to kick us off with this session, talking about the effects of neurodevelopment and the home environment on behavioural outcomes following preterm birth. So, and particular thanks because you're coming whilst on maternity leave, I understand. So thank you so much for doing it. Well,
thank you. Okay, thanks so much. Thank you for the introduction, and thank you for the invitation to speak here today. It's a real pleasure to be talking to you today about the effects of early brain development and the home environment on behavioural outcomes following preterm birth. So we know that being born preterm, and particularly being born very preterm, which is defined as birth at fewer than 33 weeks of completed gestation, aside from potentially causing brain injury and more serious neurological conditions, is also associated with an increased risk of delayed psychomotor development, a set of behavioural problems in childhood, emotional and cognitive problems, but also long-term an increased risk of psychopathology. The characteristic set of behavioural problems that we tend to see in very preterm born children involve problems with inattention, social difficulties on the autism spectrum, and emotional problems, as well as cognitive impairment, in particular executive dysfunction. And this set of behavioural outcomes that we typically see is what has become known as the preterm behavioural phenotype. Now, these adverse outcomes are thought to be the result of disrupted brain development at a really critical point in time when the developing brain is exposed to the extrauterine environment prematurely. But it's also really important to say that the effects of preterm birth, both on the brain and on behaviour, are really highly heterogeneous and also dynamic. So it's quite hard to predict which of these children are going to be the ones that show problems in childhood and later on in life. So there's a real need to understand both the neurological risk factors and the environmental impact that we can have on behavioural outcomes. So the adverse outcomes following preterm birth can, as I've mentioned, be predetermined to some extent in the neonatal brain. So here we can look at the extent to which brain structure and function in the neonatal period might be predictive of outcomes in childhood. But there are, of course, also a whole host of intervening environmental factors that really shape development in crucial ways. And that's particularly true for the home environment, and I've listed a few important factors here. One of the factors of the home environment that we're really interested in here is something called cognitively stimulating parenting. And I'll come back to what that is in a bit more detail in a few slides. But this variable is really interesting to us because it's a potentially modifiable factor that is under the control of the caregiver. So it could represent an intervention target to boost resilience for children. We know that cognitively stimulating parenting is associated with improved school success in both preterm and term-born children, and improved cognitive function in toddlers with congenital heart disease. What is less known is the extent to which cognitively stimulating parenting might impact mental health outcomes, particularly following preterm birth. So in the following, I'm going to discuss a couple of studies that look at these questions from a few different perspectives, asking the questions of how brain structure and function in the neonatal stage, and how cognitively stimulating parenting and other environmental variables impact on childhood outcomes following preterm birth. So this first study that I'd like to talk about uses data from the E-Prime study, which is a large neuroimaging study in over 500 very preterm born infants. Here we're going to be looking at structural imaging data from 384 infants who were scanned at term equivalent age. They were scanned at around 40 weeks post-menstrual age. So that's the age that they would have been born had they not already been born prematurely. These infants were then also followed up behaviourally at an age between 4 and 6 years. And here we have various measures of emotions and behaviour and cognition. And we also have a measure of the home environment here, as I've said, specifically cognitively stimulating parenting. And I'll come back to that in a moment. So first, I'd just like to explain the childhood outcomes that we're going to be looking at here. We used a range of measures that capture varying aspects of a child's temperament, psychopathology, cognition, and behavior. And these are predominantly parent response measures, aside from the Wechsler scales, of course. And what we did here was to perform a principal component analysis on all of the subscales derived from these instruments in order to reduce down the high dimensionality from all of these subscales into just a few representative components. And what we found here were two principal components that best describe the variance in this data. The first component is characterized by high loadings on issues with attention, social difficulties on the autism spectrum, and executive dysfunction. So these are really behaviors that we associate with the preterm phenotype. So we've called this the preterm phenotype component. So children who score highly on this component show more issues with attention, social difficulties, and executive dysfunction. The second component that we see here is characterized by better performance in terms of cognitive abilities and general intelligence. So we've called this the cognitive component. So we now have two separate outcome variables that are orthogonal to each other that capture, on the one hand, this preterm phenotype, more sort of psychopathology type behaviors, and a cognitive component. And these are the two outcomes that we're going to be looking at. So the first thing that we did was to look at the extent to which neonatal brain structure was associated with either of these two behavioral outcomes. And what we did here was to derive something called structural covariance networks. I've got a spelling mistake. I won't go into too much technical detail here of how we did this exactly, but what these structural networks reflect are a distribution of regions within which the regional volume covaries across subjects. So this means that within one network, so for example this one, an infant who has greater regional volumes in one area of this network also has greater regional volumes in another area of that same network. So now what we can do is extract the mean regional volume from each network for each infant. So again, we've reduced down this high-dimensional data from many hundreds of thousands of voxels of brain data to just 15 structural networks. So now we can look at whether any of the structural networks are associated with the outcomes at the age of 4, 5, and 6. What we found here was that only one structural covariance network in the neonatal stage was associated with behavioral outcome. The network that we see here is a network that really comes close to what we might call a salience network, because it encompasses bilateral inferior frontal gyri, the insular and bilateral inferior parietal and middle occipital regions, which is reflective of the salience network. And what we see here is that greater local volume in this network is associated with better cognitive performance in childhood. So this is really in line with what we know about the salience network and the insula in particular, which is really involved in many cognitive processes. The insula also filters information or relevant information for goal-directed behavior, and also mediates the dynamic interactions between other large-scale networks during cognitive processing. Next up, we looked at the home environment. And the measure that we're looking at here is cognitively stimulating parenting. Because I'm on maternity leave myself, my daughter has very kindly volunteered to demonstrate some of my feeble attempts, although I might add that teaching quantum computing is not reflective of my maternity leave, nor is it the bar that needs to be cleared for this particular scale. So cognitively stimulating parenting captures the availability and the variety of experiences that promote cognitive stimulation in the home. So this includes the extent to which the child is provided with educational toys or stimulating toys, maybe musical instruments, parental behaviors such as teaching alphabet, number, colors, spatial relations. Also aspects of the parent's own behavior, so how much the parents read at home, how many books there are at home, and the extent to which the child is exposed to family trips or museum visits, for example. And we end up with one measure that captures the extent of cognitively stimulating parenting. What we saw here was that the extent of cognitively stimulating parenting in childhood in these preterm children was not associated with the cognitive outcome, but was associated with the preterm phenotype outcome. So higher levels of cognitively stimulating parenting was associated with a lower expression of the preterm phenotype component. So fewer problems with inattention, social difficulties on the autism spectrum, and less executive dysfunction. If I just put these results side by side briefly, we see on the one hand that brain structure in the neonatal stage, particularly in the salience network, was associated with cognitive outcome. Actually, cognitively stimulating parenting was associated not with cognitive outcome specifically, but with the preterm phenotypes, so more with these psychopathology-related behaviors. And this suggests that these children might derive some meaningful benefit in terms of their mental health from access to these kind of stimulating experiences. But of course, this also leaves a few questions unanswered. So first of all, we've only looked at brain structure in the neonatal stage. So in the next study, I'm going to be talking about brain function. We've here also only looked at a sort of static snapshot of the brain, and in the next study, we're going to be looking at longitudinal changes in brain development. And this is also one sort of fairly narrow aspect of the home environment, so I'm going to be talking a bit more about a wider aspect of the environment as well. So the next study uses data from the Developing Human Connectome project, which is a really fantastic resource of neonatal imaging, which also includes data on over 200 infants who were born preterm. So these are predominantly moderately preterm. They weren't all very preterm, as in the previous study, but there was a whole range of gestational ages in here. Now, the great thing about this data set is that a subset of these infants were scanned longitudinally. So they were scanned once right around their birth, and then a few weeks later, again at term equivalent age. So now we can look at the extent to which changes in both structure and function over these first few weeks of life relate to outcome later on. And in this case, the children were followed up at 18 months of age, so a little bit younger than the previous study. There are plans to follow them up at a similar age, hopefully soon. And here we'll have measures of cognitive and psychomotor functioning, and again, behavioural and emotional problems in toddlerhood. We're going to again be looking at cognitively stimulating parenting, but we also have a measure of socioeconomic deprivation. Here we're going to be using the index of multiple deprivation, which is a postcode-based measure, which is based on 7 domains of deprivation in the area, so things like income, health, salary, education, and so forth. Again, first of all, I'd like to tell you briefly about the outcomes that we're going to be looking at 18 months. We had here the child behaviour checklist, which is a parent-rated inventory of their children's behaviours and emotions, and then the Bailey scales, which is the sort of gold standard assessment of cognitive, motor, and language development. And similarly to the first study, we ran a principal component analysis on the subscales derived from those 2 instruments, and perhaps unsurprisingly ended up with 2 components that sort of separate out the variance from those 2 instruments. So the first component that we see reflects high loadings on essentially all but the more physical aspects of the child behaviour checklist, so emotional problems, anxiety, attention problems, aggression. So we've called this a psychopathology component to the extent that you can call it psychopathology in 18-month-olds, but this is, of course, just a label that we've chosen. And the second component that we see reflects better performance on the Bailey scale, so better cognitive, language, and motor development. So we've termed this a psychomotor functioning component. And again, these are the 2 outcome variables that we're going to be looking at in toddlerhood. So moving on to the brain data that we have, this is a little bit more complicated than the first study that I showed you. We here have a measure of brain structure and a measure of brain function. And I won't go into too much detail. The brain structural measure, similarly to the first study, reflects local tissue volume in areas of the brain. We did not derive structural covariance networks in this analysis, but we're going to be doing whole brain voxel-wise analyses. And then we have a measure of brain functional connectivity. The measure that we're using here is called degree centrality, and this reflects at each voxel in the brain, that voxel's connectivity to the whole rest of the brain. So how connected on average is that voxel to the rest of the brain? So now we can look at 2 different types of effects. The first effect that we can look at is what I would call a cross-sectional effect. So this is just looking at the extent to which brain structure or brain function in the neonatal stage is predictive of outcomes in toddlerhood. But because we have this longitudinal imaging aspect, so we have scans just around the birth of these infants, and then a few weeks later at term equivalent age, we can now look at the extent to which longitudinal changes over those first few weeks of life in both structure and function of the brain are associated with outcomes in toddlerhood. So the first effect that we saw here was a cross-sectional effect in the right cerebellum, which was associated with psychomotor functioning. So what we see here is that better psychomotor functioning in toddlerhood was associated with greater relative right cerebellar volume in the neonatal period. This is really highly consistent with the existing literature on the cerebellum. So we know that the cerebellum is, of course, involved not just in motor coordination, but also in a whole host of cognitive processes. The cerebellum is also particularly vulnerable to injury following preterm birth. And we actually found that these results didn't hold after excluding infants who had major brain abnormalities. So that suggests that this association might scale with the extent of brain injury. Next up, we found a longitudinal effect of brain structure in subcortical regions on the psychopathology variable. So let me just unpack this a little bit. What we saw here is that increased psychopathology in toddlerhood was associated with a more rapid decrease over time in relative volume in the right thalamus and the subthalamic nuclei. So if we just focus on this green line here, first, these are the infants who end up with higher levels of psychopathology in 18 months of time. They showed a more rapid decrease here in the relative volumes of these regions. And just to be very clear, this is not an actual reduction in volume in these regions. They're, of course, growing over time. This is a reduction in the relative volume relative to the rest of the brain and relative to the relevant age norm. So in essence, the extent to which these regions deviate more rapidly from their age norm in terms of their volume was associated with greater levels of psychopathology in toddlerhood. We had one final result from the neuroimaging analysis in terms of functional connectivity. So what we saw here was that better psychomotor functioning in toddlerhood was associated with a more rapid decrease in terms of this measure of functional connectivity that we have, this degree centrality, in the sensory-motor cortex. So again, if we look at the green line here, these are infants with higher levels of, or better psychomotor functioning at 18 months of time. They showed a more rapid decrease in functional connectivity in the first few weeks of life in sensory-motor cortex, which might seem a bit counterintuitive at first, but we know from term-born infants that sensory-motor cortex actually does show a normative decrease in this measure of functional connectivity over the first few weeks, which we think is a sort of fine-tuning process where sensory-motor cortex is becoming more functionally specialized in terms of what other areas of the brain it's connecting to. And so we can see that early on in life, the extent to which this fine-tuning process is happening more rapidly is then associated with psychomotor functioning in toddlerhood. But then let's move on to the environmental effects that we were looking at here. So again, we had a measure of socioeconomic deprivation, this area-level measure of socioeconomic deprivation, and the same measure of cognitively stimulating parenting that we talked about in the first study. What we saw here when we looked at their association with our two outcome variables was that psychopathology was associated both with this level of deprivation and with stimulating parenting. So higher levels of socioeconomic deprivation and lower levels of cognitively stimulating parenting was associated with higher levels of psychopathology in toddlerhood. However, psychomotor functioning was only associated with the index of multiple deprivation. So here, higher levels of deprivation were associated with lower levels of psychomotor functioning. So again, we see this sort of selective association of cognitively stimulating parenting with the psychopathology component, but not with the cognitive component. And I just wanted to put this briefly before I wrap up into context of an analysis that my colleague Leila Hedaya ran on the E-prime data that I presented in the first study. So Leila ran a clustering approach in order to identify distinct subgroups of children based on their similarity profiles. So that was based predominantly on childhood outcomes, again, outcomes of cognition and behaviors and emotions, and also their neonatal variables. What she found was three distinct subgroups of very preterm children. A resilient subgroup that were characterized by more favorable childhood outcomes in terms of their socio-emotional skills and their executive functioning. An at-risk group, which had the poorest outcomes in terms of socio-emotional and executive skills. And then an intermediate group that had poorer outcomes compared to the resilient group, but more favorable outcomes compared to the at-risk group. And when we now compare these three groups in terms of a range of external variables, we see that the environmental variables are really the ones that can distinguish between the more resilient groups and those with the poorer outcomes. So what we see is that the resilient subgroup was the group that had the highest levels of cognitive stimulation in the parent, cognitive stimulation in the home. whereas the at-risk subgroup, so the group with the lowest or the poorest outcomes, were those who were actually at greatest clinical risk in the neonatal stage, so they were the infants with the highest neonatal sickness at the outset. And the intermediate subgroup was the group that had the highest level of sociodemographic risk. So we can see that cognitively stimulating parenting seems to really confer a bit of a benefit to these very preterm children, whereas sociodemographic risk can push outcomes closer to those who are actually at particular neonatal risk. So just to bring this together, we've seen that both neonatal brain structure and function can be indicative of behavioural outcomes following preterm birth, and I've highlighted here in particular the rate of brain development, so these longitudinal changes over the first few weeks of life that were associated with outcomes. But we've also seen that these area-level indicators and individual levels of individual-level environmental factors are differentially associated with psychopathology outcomes and cognitive outcomes following preterm birth. So we saw that higher levels of socioeconomic deprivation is associated both with increased psychopathology and poorer psychomotor functioning, whereas cognitively stimulating parenting is selectively related to reduced psychopathology. That, of course, really highlights the importance of the immediate family environment and emphasizes that we really need to understand both the connectome and the home if we want to understand how to help these children who are born very preterm. Because cognitively stimulating parenting is a potentially modifiable factor, it could represent a target for intervention. And there may be real scope for intervention here. There is some evidence that parents of very preterm children are less involved in the cognitive stimulation of their children. And also we know that parental and family interventions can be effective in terms of improving developmental outcomes in low birth weight cohorts and preterm infants. So we're hoping that this will bring a positive direction for future interventions. And this will be particularly important if we really want to find ways to foster resilience in these children by targeting those aspects of the home environment. So thank you very much. I'd like to thank my collaborators, particularly my colleagues at the Centre for the Developing Brain at King's, and again, my baby for letting me get off for a few hours to you here today. Thank you.
Thank you. Brilliant and clear talk about such a fascinating and important area. So it obviously makes the case for perinatal psychiatry very well as an intervention, and something that's been a huge change since I started working that now perinatal, in terms of the cognitively stimulating, the opposite side of that, and maternal depression and other issues which were modifiable. So just wondering what you thought about the sort of cultural aspects of different cultures and how different cultures might approach, the same thing, having learnt that might be useful, or how we educate parents to do, to enable them to do, provide what we think is helpful.
Yeah, I mean, that's a really good question. I don't think I have a clear answer for that, but I think there might be an element to which some of this can also happen outside of the home. So I think some of these aspects of stimulation could happen in school environments as well, so that it could be, aside from family interventions, we could be running interventions in schools or in other sort of centres that provide that sort of support. And I think the type of cognitive stimulation might be less important than a sort of variety, so I think different cultures might have different perspectives on what sort of constitutes cognitively stimulating parenting, and my sort of instinct would be that would have the same sort of effect as well, because I think since we see that the effect is more on the sort of general functioning and psychopathology, a lot of it might have to do with interpersonal communication and nurturing, rather than specifically teaching the alphabet to toddlers, for example. So I think there's a real sort of wide scope for what can be done that can fit into different cultures, I think.
Right. Thank you. I think we have a question from Neeraj. I don't know if you can get the microphone.
Hi, thank you very much for a very interesting talk. I was wondering, can we apply some of the lessons learned from studies that you've presented with people with children with preterm Can we apply the same learning to children with birth anoxia, and what are the parallels we can draw, particularly in relation to subsequent parenting, etc.?
Yes, I mean, our study obviously only had those preterm children in the analysis, so I can't make absolute statements about other groups of children, but I think at least what we see in terms of the variation of gestational age, the results seem to apply not just to extremely or very preterm birth children, but also moderately preterm and potentially also term-born children. So I suspect this is not an effect that's just specific to preterm-born children, and might be applicable to other clinical cohorts as well.
Great, thank you. Chris.
Thank you, that was a really, really lovely talk, very clear. So in our house, there's a long-standing debate about whether time is better spent reading books on quantum computing or mucking around on the football pitch. And so there's, and we know that there's evidence about physical activity, obviously, and changes in brain structure and function. I wonder whether you've looked at that as a potential confound or factor in, obviously, your first study up to 6 years old rather than 18 months, or whether there's any literature that can help us resolve this debate.
Sort of different types of activities. Yeah, I mean, the measure that we used is admittedly a pretty crude one, because it just, you end up with one. With physical, yeah, we don't have, I'm not sure if we actually have data in that sample on physical activity. That would be a really interesting thing to look at, see if it's sort of about stimulation and challenge, in particular, if it is about that sort of more cognitive aspect, which, although not to say that physical activity also has its cognitive stimulation, I suppose. So I'm afraid we don't have that data, I haven't looked at it, but that would be a really interesting thing to look at.